everyone. My name is Jennifer Ziolkowski, and I'm the CFO of Solid Biosciences. It's a pleasure to talk to you this afternoon, and I apologize in advance if I'm not able to move this along, but I'm sure someone will help me if I can't do it. Okay. I will be making some forward-looking statements today, and our legal team has asked that you read this in excruciating detail. Those are all of our disclaimers. So who are we and what are we doing? Well, at Solid Biosciences, we're singularly focused on discovering and developing meaningful therapies or even a cure for Duchenne muscular dystrophy, or DMD. We are so committed to this because we were founded by those personally touched by the disease. We've spent a lot of time working on a comprehensive approach to address all facets of the disease. And I believe we have the science and capabilities in place to develop transformative therapies, starting with our gene therapy program and our operational infrastructure. So just a little bit about DMD for those of you who aren't familiar. It is a rare disease, but one of the more common rare diseases. It has a large patient population. It is caused by one of more than 1,000 different mutations in the dystrophin gene. And dystrophin protects the muscle against contraction-induced injury. It is really important for muscle health and regeneration. It is progressive and irreversible. Sorry. It is diagnosed in early childhood, and a patient with DMD is typically in a wheelchair by the time they're in their teens. And by the time they're in their 20s and 30s, their cardiac and skeletal muscles have deteriorated so significantly that their heart is unable to pump blood, and their lungs are unable to pump air. It is 100% fatal. There's a significant economic burden associated with the disease. So we're looking for a solution for patients as well as the ecosystem of caregivers that are required to support them. Currently, there is no meaningful therapies available in the United States. The standard of care is glucocorticoids, but they have significant side effects. The one disease-modifying technology available in the United States can treat approximately 13% of the patient population. So, as you can see, there's a real need for innovative and meaningful technologies to treat all boys with this disease. Uh-oh. Oh boy. Here we go. I mentioned that we were founded by those personally touched by the disease. Our CEO, Alon's son, Atani, whose name means solid in Hebrew, was diagnosed with the disease 6 years ago. He quickly discovered that there were no treatments available for his son, and he determined that the typical drug development framework wasn't going to work. So he founded a company with the disease at the center, and he put patients at the center of everything that we do. Because of our focus, our approach addresses the full spectrum of the disease, from the underlying causes to the multiple manifestations associated with the disease. Since our inception, we have collaborated with and surrounded ourselves with experts in the scientific field and the DMD community to evaluate and bring forth only those technologies that we believed have the greatest chances of success. We organize ourselves around 4 different categories: corrective therapies, disease-modifying therapies, assistive devices, and improving our disease understanding. Today, I'm going to spend most of my time talking to you about the corrective therapies. Here's our pipeline. You can see we have a strong pipeline, and it's a big focus for us. But today, as I mentioned, I'm going to focus on talking about our gene therapy program, SGT-001, which is probably one that some of you may be more familiar with. Our lead candidate, SGT-001, is an AAV-mediated micro dystrophin gene transfer candidate. It is mutation agnostic, and based on our robust preclinical program, we have demonstrated widespread micro dystrophin expression in both large and small animal models. It has been shown to be durable, and it correlates with significant improvement in functional benefit. Our unique construct includes the latest advances in microdystrophin biology, and we've spent a lot of time working on scaling our manufacturing so that we can provide this therapy to patients. This is an illustration to show you the biology behind SGT-001. On the left is healthy muscle, and you can see the dystrophin protein is attaching to the muscle membrane and the overall stabilization of the muscle itself. As I mentioned before, dystrophin protects the muscle against contraction-induced injury. I also want to point out the dystrophin-associated proteins, which you can see in, hopefully they show up as purple and blue, yes. And those are also very important for proper muscle function. All DMD patients have a mutation in their dystrophin gene that results in the absence or near absence of dystrophin. As you can see in the center, without dystrophin, the muscles are deteriorating and those dystrophin-associated proteins are disappearing. SGT-001 delivers a dystrophin surrogate called Micro Dystrophin that rescues muscle integrity. But why Micro Dystrophin? Oh, that went fast. Okay, Micro Dystrophin, why? Well, the full dystrophin gene is huge. It's the largest gene in the body, and it doesn't fit in an adeno-associated virus, or AAV. AAV is a common delivery mechanism for systemically delivered gene therapy. Micro Dystrophin is rationally designed to fit into AAV, and we've chosen AAV9 due to its affinity for skeletal and cardiac muscle, and because it's been well characterized in other indications in the clinic. Dystrophin is complex. The top picture here, you can see the full-length microdystrophin and its multiple subdomains that support its critical functions. After decades of research, we carefully selected the regions that we wanted to retain in our microdystrophin construct. Some of those elements that we selected include neuronal nitric oxide synthase, or those elements that promote neuronal nitric oxide synthase, or nNOS activity. nNOS is critical for proper oxygenation of the muscle. We also selected a CK8 muscle promoter, which restricts expression to cardiac and skeletal muscle. Here, we wanted to demonstrate the widespread expression we can achieve using SGT-001 in our mouse model. On the top picture, you can see normal dystrophin in a healthy mouse and its expression in 4 key tissues. Underneath that, you can see in a dystrophic mouse, the absence or near absence of dystrophin. SGT-001, when delivered intravenously, results in very clear microdystrophin expression in all the 4 main tissues. We also determined that it was very important that we were able to accurately characterize microdystrophin. So we used 3 different assays with which to do so: immunofluorescence, Western blot, and mass spectrometry. And all 3 of those were used throughout our preclinical program. This is some of our preclinical data where we show that with sustained microdystrophin expression, we can show that it correlates with improved functional benefit in canines. So in the picture to the left, you can see microdystrophin expression at 1 month and sustained microdystrophin expression at 30 months. We presented this data at ASGCT this past year, and we currently have data that goes out 3.5 years. Over to the right, you can see that microdystrophin expression correlates with a dose-dependent, statistically significant improvement in muscle function. I mentioned earlier that dystrophin protects the muscle against contraction-induced injury. It also restores and localizes those dystrophin-associated proteins that I mentioned earlier called nNOS. We ensured that we included those nNOS binding domains in our construct. In this picture, you can see that 24 months after administration, what looks like healthy muscle in pink and clear microdystrophin expression as well as clear nNOS activity. And on the right, you can see that animals who were given a microdystrophin with nNOS were able to resist fatigue and run for longer than an animal with a microdystrophin without it. This is one of the reasons we believe that our program is differentiated. A little bit about manufacturing. We knew from the beginning that manufacturing would be a challenge, and we've spent time since our inception in 2013 on this. And one of the reasons that we've done that, well, there's 3 reasons. One is, as I mentioned, we are administering systemically. We hope to treat both children and adolescents, and at 5e13, our dose isn't small. So there's a lot of drug that will be needed and a lot of manufacturing required, so we've focused on that. In the meantime, our immediate intention was to supply for our clinical trial and ensure that we could do that without restriction on age and without restriction on weight and at whatever frequency we needed. In addition, we wanted to make sure that we could scale up for commercial purposes without significant changes to our process. We are currently in suspension and a process that was developed by our in-house process development team. So now I'm going to talk a little bit about our clinical trial. We started our clinical trial last year. It's called Ignite DMD, and it's a phase 1/2 adaptive clinical trial. It is randomized, open-labeled, and controlled, and it is a single ascending dose study design, which means that we are able to and plan to evaluate SGT-001 in multiple doses. We plan to enroll 16 to 32 patients, and one of the differences in our program is that we plan to enroll both ambulatory and non-ambulatory children and adolescents. Our first patient was dosed in the beginning of the year in February, and the patient suffered lab abnormalities, and the FDA put our trial on hold. The event quickly resolved, and after discussions with the FDA and changes in our protocol, the clinical hold was lifted, and the trial is currently underway at the University of Florida. The trial's primary endpoints are safety and efficacy. And just a little bit more detail about the trial, I mentioned that it was a controlled study. So patients will be randomized into either a treatment group or a control group to receive treatment at 1 year. We did this very intentionally to ensure that we could evaluate the safety and efficacy and get as much data as we could about SGT-001 in multiple patient populations. We are currently enrolling Cohort 1. As I mentioned, we're at a dosage of 5e13 vector genomes per kilogram, and we'll dose escalate as appropriate in future cohorts. We are planning to provide an update of our pre-specified interim analysis in the second half of 2019. As I mentioned, we are 100% focused on DMD. We are a really passionate group of people who are looking for a solution for these kids. We are excited to continue to evaluate our program SGT-001 in the clinic in the hopes of doing just that. We look forward to keeping you updated on our progress, and if I'm not out of time, I'd be happy to take some questions, but I don't have a timer up here. Or maybe I do. Oh, 10 seconds, anyone?